This is a special episode of the Stem Cell Podcast, ISSCR 2022, Day 4. Hey, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. Today, we're back with our final episode of the ISSCR 2022 annual meeting in San Francisco. We'll be discussing highlights from Day 4 of the meeting and sharing our thoughts on how it all went. If you're tuning in for the first time, be sure to visit www.stemcellpodcast.com or your favorite podcast app to check out our episodes covering the 3 previous days of the meeting. We're going to kick things off on Day 4 in just a minute, but before we get to that,
can't get enough stem cell science? STEMCELL Technologies has a huge selection of webinars available on demand. Visit stemcell.com/webinars to explore your favorite topics in stem cell research.
So I'm going to start by rounding up the last plenary of the evening yesterday on Day 3, which was plenary 5. There was a nice talk from April Pyle, who's a friend of the show. She's been on, showing some impressive engraftment and elaborate tools to interrogate muscle regeneration. That was a nice talk. A lot of it published, but some new. Also, Jürgen Noblich, also been on who hasn't been on the show? Jürgen from the IMBA, we all know him. He was a great guest. I love about his talk and generally his science and his class of scientists is that they're like from the old school. They apply classic methodologies, in this case, genetic screening, but appropriating all the new tech, in this case, CRISPR and organoids. He's, one of the originators along with Madeline Lancaster of organoids. So he's really ushered in the old and brought it into the new, into the future. With his classic science, with new methods. In this case, he was looking at loss of function analysis and autism spectrum genes, really to understand what compartments they affect by using this kind of mosaic mutation approach that I thought was really refined and emblematic of the most cutting edge approaches that we're using these days. Then there was Yaoqun Wang, who was talking about vascularized cortical organoids. And my only qualm there is that the vascularization was mediated by HUVEC, which I've always thought is kind of an outlier in terms of endothelial cells, not necessarily physiological. I don't know that they necessarily respond to the niche specific cues like maybe a brain microvascular endothelial cell might. But nevertheless, I mean, the real key there is if you can vascularize these organoids, you can scale them. And I think that was the primary endpoint and goal of that study. And he showed some impressive results regarding the survival and ability to grow those organoids to larger sizes. And then finally, the reason why I stayed up and got into those plenaries in the first place was to see Lorenz Studer receive his ISSCR Achievement Award, which is well-deserved. He's one of the originators of the therapy. And, not only was he the early champion of the potential, but he was, I think, one of the major mediators of reaching this point on the precipice of culminating all the efforts of neurobiologists and stem cell researchers across the globe. He had some really inspiring words to say to young scientists, investigators. I was touched and stirred by that. He had some update of his trial, the phase 1/2 trial. The safety profile looked good and feasibility of delivery looked really strong. It's just waiting for the efficacy, which I'm really optimistic that we'll see a benefit there. And what I liked here wasn't really just a tour of all his many successes. I mean, it was amazing just to see the brief review that he did in a very humble and gracious way. He's generated more than 50 cell types. We talk about the early days of trying to figure out all the cells that we could differentiate. He made about half of them. So all credit to him. But in this talk, he wasn't even reminiscing and taking a victory lap. He was talking about his unpublished work and trying to facilitate, enable maturation. It's a real problem in the field as we get these immature phenotypes. He has this gen tonic cocktail, which was a bit of a laugh. He showed a cocktail on the slide to illustrate his point. But the Gen Tonic cocktail, it doesn't just get you turned up at the party. When you're talking about iPS cells, it really does seem to have a profound effect on maturation. So that was the close for that. And then, after that reception the night before, I had to shut it down. Aruna, I'm sure you were out there in the streets, maybe at the German party that's legendary, but I was catching my Z's. What do you think, my friend?
Yeah, it was very well deserved award presented to Dr. Studer. And I did see Dr. Studer and his lab enjoying and celebrating out in the town, getting some nice dinner at a local restaurant. That was refreshing to see. And yes, I did hit up the German party, the German Stem Cell Network's annual party, which is always a lot of fun. But after a night on the town, yes, I did attend the Saturday morning roundup session, the breakouts. I had to wake up early for that, but it was definitely worth it. I actually spent most of my time, all of my time really, in a single session, which was immunological responses to fight infection and disease. A lot of really cool talks utilizing stem cell models and stem cell therapies to address different infectious disease, viral illnesses, and so on. Dan Kaufman really got things started off on a nice note with this talk titled Identification of Novel Regulators of Natural Killer Cell Mediated Anti-Tumor Responses. And this was very relevant because for some of the later plenaries that we'll get to in a little bit, a lot of his work has actually been spun into some IP that's being used for clinical trials and so on. So it's really a really cool example of true translation in the stem cell derived NK cell and iPS derived NK cell field. They're able to identify genes that, when knocked out, were able to make tumors more sensitive to NK cell attack, and really mention this CHIMP2A as a novel regulator of NK cell activity. When you knock out this CHIMP2A, it increases the NK cell mediated killing of glioblastoma, which is, of course, a tumor type that we talk about a decent amount here on the show, really severe in terms of its prognosis. But good to see that some of this NK cell work is being used to address that particular malignancy. Moving on to a talk by Kyle Lowe, who is really an emerging young superstar in our field. He's a young PI over at Stanford who has been in their stem cell institute for a while now. I've actually overlapped with him a bit during my time at grad school over there. He had a really unique talk using iPSCs to provide a new toolkit to study biosafety level 4 viruses. This is, of course, something that's relevant to the pandemic that we're in these days. SARS-CoV-2, the virus that causes COVID, is not a BSL-4 level virus, but BSL-4 viruses, just by the nature of how serious they are, and in some cases how scary they are, haven't been studied as much as we'd like them to be studied. And Dr. Lowe is presenting a cool stem cell model of Nipah virus infection, which is a virus that actually has a 60% fatality rate. And it's known to affect the blood vessels, but in terms of the cell type specific infection of the Nipah virus in the subtypes of blood vessels, it hasn't been super well understood. So Kyle is really an expert in differentiation, all sorts of differentiation. And I remember this from my time back in grad school, and they've been able to develop a really beautiful validation of vein and arterial endothelial cells from pluripotent stem cells, and basically utilizing this validated model to study Nipah virus infection. So he was able to find that the Nipah virus can specifically infect the arterial endothelial cells derived from their differentiation protocol, but not the venous endothelial cells. And it's again, I think the first time something like this has been demonstrated, and I think their manuscript should be coming out pretty soon in probably a pretty high profile journal. Next, moving on to Nadia Rajab, who was benchmarking and developing pluripotent stem cell models of macrophage biology. You mentioned Ali Moser yesterday from the Svensson lab at Cedars-Sinai, who's actually doing some similar work. And Dr. Rajab here is actually showing that proliferation is a driver of quorum sensing in pluripotent stem cell derived macrophages. This is a really hot topic, deriving macrophages from pluripotent stem cells and utilizing for different applications. Moving on to Sharon Fleischer from Gordana Vunyak-Novashkovich's lab, who actually gave a talk later at one of the plenaries, I believe plenary number 6 this afternoon, Saturday afternoon, recapitulating patient-specific autoimmune mediated heart disease with iPS-derived cardiac tissue modules. Really, the focus here was on a disease that's pretty well known, lupus, where apparently, and this is a fact I didn't know, 25% of lupus patients end up developing myocarditis or an inflammation of the heart, which can be pretty serious in a lot of situations. And do these patients have kind of auto antibodies that would specifically target the heart? This is a question that Sharon's model and system was helping to address. I think it was a really great study because it incorporated a bunch of clinical data, serum collection from lupus patients, and also an iPSC-derived cardiomyocyte model, and looking at the uptake and binding of these auto antibodies in lupus. Also looking at staying in the realm of viral infections from the previous talk, from the earlier talks that I was mentioning. Daniel Veronese Paniagua, apologies there, from Washington University, St. Louis. And he was using basically pluripotent stem cell derived islets as a model to study the role of Coxsackie virus infection, the role of a particular gene, IFIM1, in islet response to CVB3 infection. This is something, again, that I had a bit of an interest in because I've thrown this virus onto cardiomyocytes back in the day, and it's cool to see how this virus can also impact islet cell viability. Certain SNPs that apparently went overexpressed in the islets caused a stronger immune response after Coxsackie virus infection. So nice throwback to my own grad school days. Um, Andrew Khalil with a pluripotent stem cell derived vascular model to look at vascular dysfunction in COVID-19. We had to throw COVID-19 talk in there at some point, right? At the tail end of the pandemic. One thing that he showed was a differential infection of smooth muscle cells as opposed to endothelial cells in response to SARS-CoV-2, which is, of course, the virus that causes COVID. And finally, wrapping things up with Sina Bartfield looking at the infection in innate immune signaling associated with cancer in the gut. So, really nice overview of some topics that were near and dear to my heart when it comes to utilizing stem cells to study viral infections. I think this has been a very relevant topic of study that we've actually highlighted on the show quite a bit recently over the last couple of years during the pandemic. So that was cool to see the work being done in that field.
And you capitalized on that, like many others in the field, pivoting your research program a bit to, demonstrate that COVID could infect cardiomyocytes derived from iPS cells. So yeah, near and dear to your heart, and a lot of action there. I spent my concurrent session in the CA, the clinical applications um, room. And there was more talks I'm going to highlight right here, but just very briefly, there's a lot of RPE at this meeting generally. And in this session, there was a talk by Andrea Barabino, who was talking about iPS-derived RPE in large animals. And I think this is a nice complement to all the other studies. Sally Temple working in the rodent, Kapil Bharti also in the rodent, but large animals, perhaps a better model of what goes on in human, given the scale, the size of the eyes, and the proportion of rods and cones is not exactly the same in rodents. So a need perhaps to examine this, although I still think that Temple's models for showing function of the cells was better, but still impressive work from Andrea Barabino. Then I caught a talk by Ran Jing, who's from Daily Lab, a Travel Award winner, talking about how they repurposed the story that they had. They had these easy T cells, the EZH knockdown cells that they showed a while back, could improve the differentiation to definitive fates, definitive hematopoietic fates from iPS cells. Here, they were taking that capacity for improved definitive hematopoiesis and utilizing it to create this kind of off-the-shelf T cell. They call them these easy T cells, CAR Ts, that they could, this is what we're talking about. It's very thematic in this meeting, is generate a kind of founder line that then could be applied off the shelf. This one wasn't allogeneic, but I think the key here is showing that you could get efficient generation of T cells, and you could engineer them so that they were CAR T cells and they function very similar to peripheral blood mononuclear derived CAR T cells. So it looks like, at least in principle, we have a product that could be made, a CAR T cell that could be made from iPS cells. And that's a major step forward, I think, because definitive and lymphoid differentiation from iPS has been elusive. Then there was a little bit of a twist for me. There was this talk from Elias Zambitis, who's from Johns Hopkins. He had way too many slides, He only presented half of his talk, which was meant to be about ZAV 939, not just a Wnt inhibitor, was the title, and talking about all the signaling that it does besides this more, I think, widely appreciated use in the Wnt pathway. And it aligned a little bit with what Austin Smith talked about earlier in the meeting, kind of deconstructing how this compound, specifically ZAV 939, can have effects on multiple signaling um, pathways and events. But the real key to me here that was so mind-boggling is that I kind of got lost with this whole mechanism on the ZAV 939. But at the very end, as they were cutting him off and he was just trying to race to his final slide, he showed this amazing, Arun, amazing
Mm.
chimerism. Apparently, what ZAV 939 did in his specific turn maintenance, he called it turn media for maintaining, a cocktail for maintaining IPS in a naive state. They contributed like bonkers chimerism injected into mouse, like a lot of the vasculature. He just showed a Z-stack and didn't really provide, I don't think, elaborate data on the degree of chimerism. But I really wish they had given him twice the amount of time, in spite of the fact that he went too deep on the turn. I really wanted to see what was going on with that chimerism. It was really impressive. I hope it's close to publication so we can get a look at that. And then there was again this idea about RPE, right? Generating RPE, although in this case it was a wrinkle. This was from Sabia Hasibekaroglu. Sorry if I mispronounced your name there, from Andres Nagi's lab. And this was interesting to me because they were tackling wet AMD. You know, all these AMD talks begin with there is a therapy for AMD, but only the AMD that's 10% of all cases, wet AMD, and 90% of cases, the dry AMD, there's no treatment. But while there is a treatment for wet AMD, this kind of VEGF trap, this was an approach that obviated the need for repeat administration into the vitreous of these VEGF trap or other anti-angiogenic compounds. It was a means of delivering cells that then would continually generate this VEGF trap. And then they had this fail-safe mechanism as well with the suicide, and also a kind of hypogenic approach to make a universal cell line. So it was a kind of 3-in-1 approach to address wet AMD. And I thought that was very cool. And, you know, apart from other talks that I thought were also interesting that I'm not going to highlight, I thought this session was great and really emblematic of how so many groups are moving in parallel to address this translation, you know, and specifically in these hematopoietic delivery kind of constructs and RPE. So many talks about that at this meeting, Arun.
Absolutely. I mean, solid morning sessions here on the very last day. Even though it's a Saturday, we're still hearing some great science on a weekend, no doubt about that. Moving on to plenary number 6, the second to last plenary of the entire conference, which was titled Technologies that Model and Direct Emergent Cell Behaviors in Stem Cell Biology and Regeneration Behaviors. This was a nice set of basic plenary talks in contrast to plenary 7, which is very, very translational. Started things off with Alex van Oenaarden looking at single cell rope ribosome profiling. This is pretty astounding. You know, the fact that you can do ribosome profiling on individual cells, and not only that, couple that with single cell RNA sequencing. I think this is poised to become a very, very powerful tool in the stem cell biologist toolkit. Moving on to Smeata Krishna Swami, who unfortunately had a couple of technical issues with her talk. But this is a little different from what we were used to. A lot of equations, a lot of math being thrown on the screen. I haven't taken math in a few years, so it was a little intimidating to me, but still a very powerful computational analyses that she was presenting. She's a computer scientist at Yale, who's actually really interested in denoising single cell data sets, and was also looking at the deconvolution of calcium signal dynamics, which is something that's near and dear to my heart, certainly. Moving on to someone who doesn't need that much introduction, Gordana Vunyak-Novakovic from Columbia, whose trainees and papers we have covered extensively on the podcast, including some of the work that she presented in this particular talk. Of course, she was talking about some of the work of Casey Ronaldson Bouchard, some of these organs on chips and multi-organs on single chips, including the Nature Biomedical Engineering paper that Casey just published, where they're able to combine skin, heart, bone, liver on a single chip, and with a complex vasculature, complex cell types, complex media compositions that were in a single chip. Also, Naveed Tavakol was highlighted here, also a trainee in Gordana's lab, a former guest of the podcast, talking about his engineered human bone marrow with hematopoietic properties. This is an upcoming paper, an upcoming project of his, and a lot of other projects in Gordana's lab. So many cool projects going on over there, modeling ischemia reperfusion injury in a sex-specific IPSC context. Just so many cool things happening over there in Colombia in Gordana's lab. Also, a breast cancer metastasis model using this multi-organ chip. That's also super cool. Another talk that I thought was really astounding and very different from what I was used to. This is Satoshi Toda from Japan, a very, very cool synthetic bio talk. Not a ton of pure synthetic bio talks at this particular conference, but here he was looking at programming of multicellular pattern formation with these synthetic cell signaling model systems, and in particular, a unique synthetic bio model where he was using GFP itself as a synthetic secreted morphogen. Really astounding, kind of mind blowing concept here how GFP and also a synthetic notch receptor can be used to modulate and examine, morphogen gradients in a very simple pluripotent stem cell model. Something very different, but very cool to see. I know a lot of people were paying very close attention to that one. And then finally, Joanna Wisaka from Stanford, who was presenting the her talk, which is actually the ISSCR Momentum Award lecture, from nucleosomes to human faces, her journey in stem cell biology at Stanford and over the years. She's really an icon in chromatin biology and basic stem cell biology. And also it has developed an evo-devo evolutionary development aspect to her work. A lot of beautiful work using cranial neural crest cells to study human evolution, in particular primate evolution, is, I think, very well deserved. She's been an icon in the field, and I'm sure the years to come are just going to have so much more great work coming out of her lab.
Yeah, and she's already really changed the game. And just, I really respect and appreciate her mind because she's one of those unique scientists who just is able to think along different directions. I remember when she came out with these really, I think now appreciated as seminal studies, it was, I don't think anyone was thinking in that box. And she did a couple of things. One, she came from this landscape of epigenetics and understood the tools and the importance of those regulators. But then she was able to apply it into a pluripotent stem cell system, which wasn't really her wheelhouse at that point. And then to take it in, as you said, Arun, into a direction of Evo, Devo, and talking about like craniofacial development, which I think just putting all those things together in one box is really amazing. And my takeaway there was, yeah, it all comes down to the face, is what Joanna Wisaska was pretty much saying, the transcriptor factor dosing that's dictating the phenotypes. So, now I have some explanation for this humongous forehead. I can't really, couldn't explain it before, but now I guess my forehead dose was pretty extreme. I got my parents to blame for that. But it was a nice, I think, short break there, which was, I think, the final break of this conference, I think, is worth noting, Arun, because it was a short break, so people didn't really have time to get away. And the whole plenary session there was out in the little refreshment break, and everyone had one last chance. Of those who remained, they had a last chance to, press hands and talk about how great it was to come together. That was really nice. Final parting there before we got back into the plenary to hear some words from the incoming president, Hai Fan Lin, who I thought importantly in emphasizing and voicing the society's goals, really touched upon how we need to integrate the global community. We need to recruit more Asian, Latin American, African people from the global stem cell community and integrate them with the society so we can get some, a more diverse group at this meeting and in this society. And of course, that's going to contribute to more great science. Then it was on to Donald Cohen. You might call it a puff piece, I guess, but I really got some tears in my eyes. Or was that you, Arun? Was it you crying or was it me who was crying? I think it was you who
It was
was
both
crying.
of us.
Yeah, I think there was a little bit of tears in both their eyes. It was a great just story and something I think we all need to hear from time to time. about how these measures that we take, and we only really see them in the lab for the most part, how that once they get out of the lab, they can really change lives. In this case, talking about bubble baby disease, is ADA SCID, and revealing some of the stats that I wasn't aware of. 50 babies have been cured of this disease, and that's a cure, going from uh, no life to just a normal life. And that was illustrated in this case by a conversation that was had between Dr. Cohen and a patient, talking about her baby daughter who was treated at a very young age and is now 10 years old, living a really normal and rich life, going from a bubble to a surfboard. There was some really nice pictures showing how she was able to, notwithstanding all the fear and the trepidation regarding a new therapy, she was pretty much untouched by that. And even, contracted COVID and was able to respond to all her vaccinations, living the life that my kids are living. So that was really nice to hear. And I think all of us in that room need to hear that from time to time. So that was great. And a great segue then to Luigi Naldini, who's, amongst one of the godparents of gene therapy, perhaps the real icon behind it all. He spoke a lot about all the measures that they're taking in his group and many groups to improve genetic engineering, to make it safer and easier, to make it cheaper, to make it able to apply to a lot of diseases, perhaps less serious diseases where the cost benefit is not so complicated. Um, so that was great. And I can just imagine what it must be like. I can only imagine. I would love to one day know what it feels like to do something your whole life and then trail a talk where your developments have saved a life and made a life. So that must have been very gratifying for him, and I think the whole group. Then it was on to Masayo Takahashi, who is also another icon in the field, talking about her work with RPE. She was, almost a decade now ago in 2013 that trial of iPS derived RPE was launched in Japan. Although it's been a bit of a rocky road, I think there was a little bit of spin, I would say, in her presentation in terms of how the course of those trials have gone. As some of those listening may remember, they went from an IPS only to a bank to an allogeneic. So those trials have progressed, and I think that there have been major challenges that emerged for all of us. But I think now, I guess doctors Temple and Barty, Kapil Barty and Sally Temple, have really caught up there, and I'm betting on them, frankly. But there's still a long way to go, and there's room for everybody to make a major contribution. And Takahashi certainly is one of the founders there. So it was nice to hear from her. Then there was I think, a really interesting story from Bob Valamir at Fate Therapeutics about off-the-shelf T and natural killer cells. And the thing here that was, I think, great, was his quote. He said, "5 years ago, this was a cartoon. Now we're closer to reality." And that is the idea of generating a cell bank for therapy, a master cell bank, and the method that they use, which I think is has benefits and costs, is this clonal expansion. They get a single cell from one cell to 100 million, and that's their master cell bank, so that they can qualify it. Although, just a note, that's 25 population doublings. And the idea there, the rationale, is that you'll have a clone. I would argue that maybe in those 25 population doublings from 1 to 100 million cells, there may be some heterogeneity that emerges. But I think it really doesn't matter in this case. Maybe you could argue, because this is a kind of mercenary therapy, I like to call it, where you generate cells that you don't need them to engraft. You need them to go in there and kill some stuff. In this case, the NK cells and the T cells are there to target cancer. You just wonder about hematological malignancy from the actual graft. So I think you got to have some countermeasures there. I'm sure they've thought of that. Regardless, I think an impressive methodology. They've really scaled up and gotten to a point where they're ready commercially to apply these cells. So that was impressive and exciting. And then finally, Arun, I guess we're going to speak about this a bit, you and I both, but there's a story we've all been waiting for, and that was kind of forecast by Doug Melton early in the meeting from Gary Menninger. from Vertex, where they injected cells into the portal vein with immune suppression. They had a series of 2, then 5, then 10 patients. The first 2 patients, a safety dose, which is half of the target dose. And they've got both those patients they've delivered cells to, and they have a third patient that they've delivered at the target dose. Primarily, the data was focused on that first patient because the second patient is only out to about day 150, or that's all the data was shared out to. But that first patient showed really, really remarkable response with effectively looks like a complete cure, is insulin independent and has a normal H1C and glucose monitoring, continual glucose monitoring shows a totally normal profile with very mild adverse events. And this was the patient that we read about in the Times. So we already knew, I guess, that this was going to be a virtual cure. The second patient, not as impressive. The third patient, we didn't really get any data on. But I would say I'm optimistic, as is the stock market. If you look at the ticker, Vertex is pumping right now. But I'm optimistic, even though there's surely going to be some heterogeneity amongst the population of patients that receive this therapy. Arun, pretty exciting stuff. What do you think about it?
Very exciting. And I think there's a reason why they have these major clinical trials wait until the very end of the ISSCR to announce some of these results, just to keep people interested, keep people in their seats, make sure people stick around for the entire session and the whole ISSCR. And of course, we also have to mention Priscilla Chan, who gave a keynote address at the very end of that plenary, talking about just in general the great work that the biomedical research community as a whole is doing, and also that the ISSCR has done over the last 20 years and bringing therapies to patients. But yes, I think the focus really here was on the phase 1/2 early clinical trial results from Vertex, hinted by Doug Melton very early on in the ISSCR. And yes, the patient 1 results were absolutely astounding. We did get a reflection of that in the New York Times late last year, but it's good to see the clinical data, and it does correlate with what the Times piece did mention. This individual had an astounding response to this therapy. Patient 2 is certainly not as impressive, but I think this is enough to push this trial down the road. Very, very good to see. And moving on to, at the earlier talks from that plenary, I think were also very refreshing. I think Don Cohn's patient advocate talk with Alicia Vicaro talking about her daughter, I think we all needed to hear that every once in a while. We needed to hear that the work we're doing is having a positive impact in the community, in the patient population, and potentially in this situation leading to cures. This is a story that was really personal, very touching to me as a new parent as well, seeing a child who is afflicted by this very serious deadly disease, and now in all circumstances, for everything else, is quite healthy, surfing in Hawaii, as was mentioned in that particular session. But yes, I think the real beauty for this session to me was to see that these iPSC derived clinical products and therapies are really reaching trials, and they're having efficacy in a lot of situations, especially for the case of that one Vertex patient, that one iPSC derived islet patient. I think Fate Therapeutics is doing a great job. Blue Rock is on the road as well. Vertex is down the road. So this is happening. This is really exciting. And especially for, I think, the trainees out there who are in a generation of trainees who are seeing these things go into patients. And, a generation ago, we were just worried about making these cells. And now we're actually bringing them into people and having efficacy. I think it's so incredibly refreshing and powerful to see. And that's it. That's all we got for this year's ISSCR 2022. I mean, we'll leave you with some of our parting thoughts in terms of the big picture of the conference, both the science part of it, the social part of it, starting with the social part of it. And this is something that every single person in our one-to-one delegate interviews mentioned. They just wanted to see people in person. Okay, this was refreshing. We're social creatures, human beings. We are. It's no doubting that. And to see everybody in person, face to face, seeing old mentors, old friends, folks who are avid listeners of the show and have appreciated the work that we do here on the podcast to bring the science to you. And just that was just really heartwarming to see that people are listening to what we say and valuing what we say. So that was great. Thank you, everybody, for taking a close listen to this year's ISSCR roundups and also just listening to the podcast in general. And when it comes to the science results, the things that really stuck with me were one, these hypoimmune talks. Okay, this is coming up again and again. The idea of shielding cells, shielding therapies from immune attack. And I think perhaps combining that technology with some of the clinical trials in iPSC derived RPE, iPSC derived islet cells, I think that's really going to take these cell therapies to the next level. Also, the power of 3D models, next generation models, whether it's Gordana talking about her multi-organ chips or tons of people talking about assembloids, which are not just limited to Sergio Pasca anymore. A lot of people across the world are adapting these next generation technologies to really great use and great application. And of course, the clinical trial results, in particular highlighted by Vertex. That was a defining moment, I think, in this field to show the efficacy, the astounding efficacy of iPSC-derived islet cells for treating type 1 diabetes, and can't wait to see what the future holds.
Yeah, man, these therapies, they're working. It's so amazing. And I can imagine this must be beyond the wildest dreams of a lot of the investigators who were at the meeting, the first meeting 20 years ago. So it's a nice, I think, 20th anniversary for the ISSCR. As Arun touched upon there, we're moving forward in terms of, our ability to approximate human embryogenesis, and we're using that for basic insight as well as for therapeutic application. It's such an exciting time to be alive and to be at this meeting. My final thought, it's a little bit out of left field, but I just noticed every talk I went to, this is not even a joke. It's a little bit funny, but it's not a joke. About 3-4 rows away from me, every time, Lee Rubin. I'd look up, I'd see Lee Rubin right there. So there's one or two possibilities here. Either Lee and I have the same instincts about what sessions to go to and where to sit in the room, or Lee Rubin, you're stalking me. And if that's the case, my friend, and you're listening now, please come be on the show. All you had to do was ask. And if it's not the case, if it's the former, I think I'm in the right company there. Lee, you got the right instincts like me. Let's get together on a collabo, maybe put something together for ISSCR 21st anniversary. But we'll be back before then with another episode. But that brings us to the end of our ISSCR 2022 episode series during the meetings here. We've met a lot of people, had a lot of fun, and we already can't wait until the 2023 meeting in Boston, Massachusetts. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com. You can also reach out to us on Twitter at StemCellPodcast or via email at info@stemcellpodcast.com with feedback. As Arun said, to all you guys that we met and that are fans of the podcast, it's been so great talking to you and, getting that fandom. I mean, we need it. As far as we know, we're talking to each other, but it turns out there's a few out there who like the show. We hope we can continue to bring it to you, and we will have another episode for you very shortly. Thanks again for listening. Bye-bye.